Oke, terima kasih sekali Mbak Nadia sudah meminta saya sebagai moderator di guest lecture from Miss Taru Marja Martinen. Yeah. Taru is from Helsinki University, a partner of microbiology. She's just finished her master.
Next month.
Oh, next month. Okay. Wisudanya next month. Yesterday you have the seminar for
my master thesis presentation seminar. The results.
Yep. So far we have a collaboration with Helsinki University, especially on antimicrobial resistance. This topic is really multidiscipline, interdiscipline, and under the organization of One Health, taking care of the animal and the environment. And we are in Faculty of Agriculture, of course, dealing with plant, which is included as a part of the environment. So this AMR topic is quite important now, and urgent to be paid attention, because many infectious diseases now becoming drug resistant. So, Taru, we are looking forward for your presentation. That must be interesting. So please, you have 1 hour or 3 hours?
Kind of maybe 3 hours.
Okay.
Yeah, thank you.
Yeah. Okay.
So about my background, I graduated or I did my bachelor's degree focusing on biosciences and biochemistry, and I graduated 2021. And yeah, I'm still currently in master's program in microbiology, biotechnology in University of Helsinki. But as said, I'm going to graduate next month when I go back to Finland and need to do one book exam. I started at Molecular Environmental Biosciences Research Group at the beginning of this year, and I did my master's thesis in that group, and I'm going to continue being a part of that group and start my doctoral studies soon. So here's the content of my presentation. So I'm going to give you a short introduction about antibiotic resistance, and also tell you something about the antibiotic producer's role in the resistance, and then tell you about streptomyces. Then I'm going to continue with the case study, which is my master's thesis results, and also I'm going to tell you what materials and methods I used, and try to explain you how you can also use them. And then I'm going to give you a short discussion and conclusion. Before I start, here are some terms and abbreviations used in my presentation. So when I'm talking about actinobacteria, I'm meaning the antibiotic-producing bacteria, such as streptomyces. And when I'm talking about proteobacteria, I'm meaning the pathogenic bacteria, like E. coli. And I'm also using the term clinical isolate, which just means that the bacteria has been isolated from clinical settings, such as like hospital. And of course, these actinobacteria and proteobacteria include other bacteria that are not antibiotic producer or pathogens. But yeah, in my presentation, I'm going to mean those. So I'm going to use a lot of abbreviation ARG, so antibiotic resistant gene, and they confers resistance to antibiotics. And also I'm going to use NGA, so mobile genetic element. And if you are not familiar with mobile genetic elements, they are plasmids, transposomes, interons, and other that kind of things, and they can move around within a genome, or they can be transferred from one species to another. So antibiotic resistance is one of the most challenging health threats of our times, and it is threatening to lead us into a so-called post-antibiotic era, meaning that bacterial infections may no longer be treated with antibiotics. And even though antibiotic resistance is ancient and natural phenomenon, it is evident that the use of antibiotics is one of the major drivers in the spread of the resistance. And agriculture and livestock production are one of the biggest sectors using antibiotics. And it is estimated that up to 90% of the antibiotics used are excreted as urine and manure, and mostly unmetabolized. And this can create a selection pressure to antibiotic-resistant bacteria, which can then lead to their spread and enrichment of antibiotic-resistant genes in the farms and surrounding environments, for example, using manure as fertilizer. In addition to containing those antibiotic-resistant bacteria, organic fertilizers, such as composted biosolids and manure, are materials in which antibiotic-producing bacteria may thrive. And what is the problem with the antibiotic producers is that they usually have resistance genes that act as a self-protection against the compounds they produce. And in addition, they can have ARGs obtained horizontally from other bacteria. Also, it has been noticed that antibiotics and other growth promoters can increase the co-occurrence of ARGs and MGAs in big manner. So this study was done in 2021 by Dr. Johanna Muurinen et al. And if you look at these figures, you can see that in this non-treatment control, there is the resistant genes and there are the mobile genetic elements, but they are not linked to each other. If you compare to this one, that antibiotic has been used, you can see that there's so many of these links. And now we are wondering that can antibiotics produce? Yeah, it's not working. Oh, yeah, no. Okay, no. Hello. Yeah. And so, okay, maybe I will change my mic. Yeah. So can antibiotics produced by microorganisms also have an influence of this ARG movement? So as early as 1973, this called producer hypothesis was proposed by Benvis and Davies, according to which some of the ARGs present in pathogenic bacteria are derived from antibiotic producers, as they found ARGs in clinical isolates that was similar to those of antibiotic-producing bacteria. And in 2017, Young et al. Found further evidence for this hypothesis, and also they showed a possible mechanism how these genes can be transferred. So it is well known that proteobacteria can transfer their DNA by conjugation, but this has not been described so far in actinobacteria. Also, in theory, proteobacteria would take up free DNA from the dead actinobacterial cells by natural transformation. But as the DNA sequences between proteobacteria and actinobacteria are different, there is low probability for this to happen. So how does the actinobacteria transfer their DNA to proteobacteria? And one possible answer is this carry-back model. So in this carry-back model, first, the proteobacterial DNA is conjugated into actinobacterial cell. And in the actinobacterial cell, the actinobacterial DNA, for example, resistant gene, can be recombined with the proteobacterial DNA by a mobile genetic element, for example, transposon. And then this sandwich structure is formed, where you can see that the actinobacterial DNA is kind of inside the proteobacterial DNA. And this DNA structure can then be released from the actinobacterial cell by cell lysis. And nearby proteobacterial cell can take up then this sandwich structured DNA through natural transformation, and then it can be added to the proteobacterial genome via homologous recombination. And now a short summary what I just told you. So it has been noticed that some of the ARGs in proteobacteria have been originated from actinobacteria, and now we know a possible mechanism how this gene transfer can occur between the bacteria. And it needs the actinobacteria and proteobacteria to be in the same environment, which is the case with manure or can be the case with manure and other organic fertilizers. And in addition, it needs mobile genetic elements for the gene transfer. And also, it is known that antibiotics and other growth promoters can increase the co-occurrences of the ARGs and MGAs. So what about the antibiotics produced by microorganisms? So now I'm shortly going to introduce you Streptomyces, the focus of my thesis. So Streptomyces is the largest genus of the phylum Actinobacteria, and they live in soils and sediments around the world. And they are gram-positive, aerobic and filamentous saprophytic bacteria, meaning they are able to degrade organic debris such as cellulose and chitin from plants, fungi, and other organisms to obtain nutrients. And they have a large linear genome with high guanine cytosine content. And usually, bacteria that have high guanine cytosine content in their genome tolerate wider range of temperatures and have higher optimal growth temperature. And for example, and this is a common feature in environmental bacteria. And also the large genome makes it possible to carry more genes and thus make it possible to adapt faster for the changing conditions in the environment. So Streptomyces has this complex life cycle during which they grow as a branching hyphae and that forms this densely packed substrate nucleum. And this substrate nucleum often penetrates deep into the surrounding substrate. And the substrate nucleum develops a specialized reproductive structure, aerial nucleum, which rise from the colony into the air. And Streptomyces spreads to the spores, and they are formed from the aerial nucleum. And the spores can survive long periods of time in challenging conditions. And when the spore encounters good growth conditions and nutrients, it germinates and starts to form the vegetative growth, so the substrate nucleum. And the production of secondary metabolites, such as antibiotics, happens typically at the same time as the transition from the vegetative to aerial growth. And these both are triggered by stress, nutrients running out, and other signals. And this life cycle of Streptomyces varies between species, but it is typically from 3 to 10 days. This is picture for my thesis work, but it's just here for example. So Streptomyces usually have many different metabolite biosynthetic gene clusters. So these that you can see, this whole line presents the genome of one isolate, and these boxes in here are the gene clusters. And because they have so many different metabolite biosynthetic gene clusters, they have the ability to produce a wide selection of secondary metabolites. And the mean number of these clusters is 30 per species. And many of these clusters have antibiotic, antifungal, antiviral, and anti-cancer activities, but also many others that include such as pigments, odors, iron transport, and osmoprotectants. And these clusters often contain the resistant genes that act as a self-protection against the compounds they produce, which makes antibiotic-producing Streptomyces a potentially important source of antibiotic-resistant genes. So now we are going to move on to the case study. So first, some background. So in Europe, Mainly imported phosphate mineral and energy-consuming nitrogen are used as a fertilizer. However, the European Union is aiming to move more environmentally friendly fertilizers, such as manure and other organic materials, from nutrient-rich side streams. But as I said before, the use of manure and composted biosolids poses the threat to disseminate antibiotic-resistant bacteria and ARGs, but in addition, they are ideal habitats for antibiotic-producing bacteria, such as Streptomyces. And this leads us to my thesis subject, which was Streptomyces isolated from bio-based fertilizers, characterization and detection of genes for antibiotic resistance and production. So the big question behind my thesis was, do antibiotic producers play a role in dissemination of antibiotic resistance? And to enlighten their role in antibiotic resistance, I studied the genomic characteristics of bacterial isolates from bio-based fertilizers, focusing on what they are capable to produce in theory, and do they contain antibiotic-resistant genes, mobile genetic elements that are needed to mitigate the resistance. Also, I studied the cultural characteristics of the isolates, so in addition to giving a starting idea of their role in resistance, my results should help guide future studies. So the bacteria were isolated from 2 bio-based fertilizers for my thesis by my supervisor, Dr. Johanna Muurinen, and master student Kirill Kotkanov. And the BBF42 was made from digested separated pig manure slurry at the pig farm in Finland. And the BBF45 was a commercially used fertilizer, which was made also in Finland, and it contained composted bio-based food industry byproducts, feed and food chips, among other things. And also, one bacterial isolate from a latrine sample taken during archaeological dig in Finland was included in the DNA extraction and follow-up studies. And if you are not familiar with the word latrine, it means like toilet. So the sample was maybe some feces. So the isolates that were initially identified as Streptomyces were stored at +4 as pure culture plates and also at -70 degrees as glycerol stocks. And isolates from pure culture plates were grown on these 3 different solid media at 2 different temperatures, so at +28 and +37, until good visible growth was seen. And from these plates, a selection of colonies were inoculated in the 3 liquid media. And the 3 liquid media were incubated at +28 degrees for 2 days with some shaking, and then at room temperature for 4 days without any shaking. And antifungal and antibiotic against gram-negative bacteria were used for selection, so to prevent growth of unwanted bacteria and fungi. And the DNA was extracted with this easy powerlyzer power soil kit by VN, and the extracted DNA was stored at -20 degrees until whole genome sequencing. So first, shortly about my cultural characteristics. A formation of substrate and aerial nucleum was seen. And these could be separated by color, texture, and elevation. And these figures A and B did not grow in any other media tested than the Gaussian number 1. And isolates from the BBF45 grew equally well in all tested media. And this one isolate here preferred ISP7. And if you look this figure F, you can see this visible branching filament, and they were formed after a few months of storage at +4. And these were also seen in other places, but not as clearly. And except for this one isolate, they all grew 24 hours faster at 37 degrees. So, we third used solid media, ISP6, isolates from big manure slurry sample, so BBF42, did not grow at all, and isolates from the composted biosolids sample, the BBF45, grew this kind of uniform bacterial mass, and the single colonies were really tiny. And on broth cultures, some small flocules, or these larger clumps, were formed, and even if the tubes were shaken, this did not mix with the broth. And this is typical characteristic for streptomyces on cultures, and it is recommended to use some sterile springs or glass beads during the cultivation to break down those nuclear bonds if there is a need for tubic liquid culture. Yes, so now I'm going to move on to this bioinformatic part of my thesis. So whole genome sequencing of the extracted DNA was done using PacBio, and it is a long-read sequencing technique. The possible adapters were removed from the raw reads, and the reads were assembled into contigs. And the quality of the adapter filtered reads and the primary assembled genomes were checked. And these primary assembled genomes, so the contigs, were annotated against different databases to get an overview of genome and see the relatedness of the isolates, and also to perform the taxonomic assignments. And the presence of ARGs, plasmids, mobile genetic elements, and antibiotic-producing gene clusters were studied by performing nucleotide blast search of the isolate sequences against these databases. And so the whole genome sequencing, the whole genome of one selected bacterial isolate is sequenced, and it produces these raw reads that it needed to assemble into a context to study them. And the goal of the assembly would be to build up the genome of the isolate as it was before cutting it into pieces. But of course, it is not so simple as you can think to process as paper going through the paper cutter. So it is quite hard to put the pieces back together the same way as they were before to form the same sentences that were in the paper. But luckily, we have some programs to do this assembly, so we don't need to do this manually. And this is modified from Melina Markkanen's presentation, who was with us before here in Indonesia, but has already gone back to Finland. So shortly about the long-read sequencing, it produces highly accurate long reads. And because of that, the genetic environment of the genes can be studied. And when I said that the adapters need to be removed before assembly of the sequences, is it because in this sequencing technique, they add these short sequences in your isolates sequence. And during the process, they should be removed already before they send the sequences to you, but there's always the possibility that your reads still contain these adapters. That's why we will do it again, just to be sure that our results are not affected with these adapters. And if you are not familiar with the word contig, it means a set of DNA sequences that overlap in a way that provides a contiguous representation of the genomic region. So these short reads can be assembled to these larger sequences, so the contigs. And I used sequence alignment a lot during my thesis. So if you're not familiar with it, it's the process of comparing and detecting similarities between biological sequences, such as DNA. So in here you can see that these two sequences can be compared, and here the sequences are similar, so they are matched. And in here, the base can be missing, or it can be some other, so then there's a gap or mismatches which lower the sequence similarity. And so the results of sequence alignment tell you how similar the sequences are in relation to each other, or you can use it to see how similar your sequence is with some sequence from the database. And one commonly used bioinformatic tool for sequence alignment is BLAST. So it comes from Basic Local Alignment Search tool. So I used this command line tool on my computer, which looked like this, but I'm not so expert with this, so I'm not trying to explain this to you because I just might to confuse you even more. But yeah, I wanted to just show you which kind of environment I did my bioinformatic analysis. And yeah, you write this kind of codes, and then it is processed in this supercomputer called Bukti. And then you can analyze huge data sets that you can't analyze with your own computer, but you need license to use these software, so they are not free.
Yeah.
But I have included these links to my slides. So if I found the program I used in the command line tool from online, so some of these can be also used as online versions, but there are size limits when you are using them. So for example, to my isolates, their genomes were too big to be analyzed with the online versions. But if you have some smaller sequences, they might be able to analyze in online versions. And I'm going to share these slides to you, so the links are available to you then. So the genome assembly quality was checked with this software WAST, which uses other software for visualization. So as a result, I saw that all of the isolated genome characteristics were typical for streptomyces, such as they had large genome from over 7 million base pairs to almost 11 million base pairs. And also they have high guanine cytosine content with average of 71.8%. And each of the isolated primary assembled genomes has variable number of contigs from 1 to over 100. And there were one large contig and variable number of smaller ones. And the N50 value that also is calculated by the software means as the sequence length of the shortest contig at 50% of total assembly length. And for my isolates, the N50 number was the same as the longest contig, indicating that my assemblies had good quality. I also did genome analysis with Faperta that can be used for genome annotation of bacteria. So it gives you overview of your input sequence and also recognizes protein coding genes, open reading frames, origins of replicon, and many other things by comparing your sequence to database. But I'm not going to talk more about this because I also just looked this to get some overview. So this Kuliah can be used as a command line tool only, or at least I didn't find it in the online. So it compares your input sequences between each other, or you can create some database if you want to use it to compare your sequence similarity some downloaded ones. So Kuliah compared the isolate sequences and counted how similar they are, and it produces this kind of nice figure where you can see the results. So if the sequences shared a similarity or the average nucleotide identity between over 95% with each other, the cells are red. So you can see, for example, BBF45-4 and BBF45-4. So it's the same isolate, of course they are similar, but also this isolate was similar with some other isolates, which was the case with also these two isolates and these three isolates. And only two of my isolates had unique sequences. And Kuliah also creates these dendrograms that suggest the possible phylogeny of the isolates. However, in my case, they should not be used as I did not use any reference species to which to compare the phylogeny. So, yeah. So next, I did the taxonomic classification using this genome taxonomy database toolkit, and it's not available online, but it includes almost 400,000 bacterial genomes and over 80,000 bacterial species to which you can compare your isolates. So, as expected, all of my isolates were classified into streptomyces or to be streptomyces, and also they had quite high sequence similarity with species they were classified into. So then I did the blast search of the isolate sequences against this FlashFinder database. So again, it compares the isolate sequences to the database. And this database includes thousands of antimicrobial resistant genes. And here you can see the resistant gene. Here's the antibiotic class resistant gene confers resistance, and the isolates are down here. And if the cell is red, the gene is present, and if the gene is absent, the cell is blue. And the numbers inside the cells, you don't really need to see them so closely. It just tells you how similar the gene of your isolate was with the database's one. So as a result, a total of 40 different ARGs were found, and per isolate, the amount of ARGs varied from 17 to 30. And most of the ARGs conferred resistance to beta-lactams, macrolides, and tetracyclines, but also genes conferring resistance to aminoglycosides, glycopeptides, tanningols, and quinolones were found. I did not use this database, but also this could be used to find resistant genes. So, but here's also the size limit that your sequence can be only 5,000 nucleotide or amino acids long, or then you can have 20 megabyte size file. But I just wanted to include this here so you can maybe want to try this one also when trying to find resistant genes. So no plasmids were detected when I did the blast search against the plasmid finder database. However, further study is needed to verify that the multiple contigs, for example, present in some of the isolates were not plasmids, as the problem with the database is still that they does not include all plasmids, and also the isolates can have novel plasmids, so new plasmids that are not yet found. But however, the isolates had in total of 7 MGA-related sequences in their largest contig. And in addition, 2 of the isolates had MGA-related sequences in their smaller contigs. suggest that these smaller contigs have chance to be mobile, but I still need to study what the smaller contigs are and do future analysis. So the secondary metabolite biosynthetic gene clusters were analyzed with on this MASH webpage version, and this is an example what on this MASH results can look like for one of my isolates. So it shows you in which region of the your isolate sequence the possible secondary metabolite gene cluster is, and also it shows you the most similar known cluster if there is one, and also how similar the cluster is. And from 15 to 29 potential clusters of secondary metabolites were identified in the isolate genomes. And these clusters, most similar end products, have many different activities. The isolates from the BBF42, so the big manure slurry, had more secondary metabolite gene clusters than the isolates from the BBF45, and the isolate from the archaeological sample had the most secondary metabolite gene clusters. However, the secondary metabolite gene clusters from 1 to 6 most similar predicted end products were known to have antibiotic activity. But also the other natural products of these clusters may have antibiotic activities, but at least they were not considered antibiotics at the moment of my thesis. And even though the ancient isolates had most clusters, only one of them was known to have antibiotic activity. So then the discussion. So the cultural characteristics were typical for Streptomyces given their life cycle. And the isolates were considered as mesophytes because of their growth temperature. And it has been seen in previous studies that a wide range of temperatures typically supports good growth of Streptomyces. However, it has been noticed that even though Streptomyces can grow on many different temperatures, the temperature for secondary metabolite production is more specific, and usual optimum temperature for the production is 30 degrees, but also, of course, this varies between the strains. And several resistant genes and mobile genetic elements were detected in the isolates. So in theory, the mobility of ARGs is possible, but the genetic environment of ARGs should still be studied to confirm their mobility so that they are associated . So if the ARGs are associated with mobile genetic elements, it could be that the production of antibiotics has induced the integration of ARGs and into MGAs. Interestingly, the isolate from archaeological samples seem to possess similar ARGs and MGAs with the modern isolates, indicating that there is already residue of ARGs in the environment, which might have ability to be mobile. And the ancient isolate is estimated to be from the 18th century. So it has the resistant genes, even though it's from the time before humans has discovered antibiotics. Also, as the isolates from the big manure slurry and the ancient latrine sample had more secondary metabolite biosynthetic gene clusters, it could indicate that the isolates being in manure or stool were likely more competition. They also produce more metabolites to have an advantage of the competition and to protect themselves. But this hypothesis should be still confirmed by studying more bacteria and genes from these samples, for example, with metagenomic sequencing to see if this really is the case, as I had so limited number of isolates. So, yeah. And the results of my study suggest that antibiotic producers play a role in maintaining and possibly even transferring resistant genes in composted biosolids. And although this was only a preliminary and very limited study on this topic, it suggests that antibiotic producers can have contributed to the dissemination and evolution of antibiotic resistance. So, investigating the co-evolution of antibiotic producers and antimicrobial resistant bacteria should be the interest of scientists in the future, and perhaps understanding the interactions of the two could open new pathways for controlling the spread of antibiotic resistance. And I want to thank my supervisors, Dr. Umum, doctoral student Elina Markkani, and Professor Marko Virta, and also CSC for making it possible to analyze my data, and also the person from DNA sequencing and genomics laboratory for sequencing my isolates genomes, and also for Kirill Potkanov for isolating the bacteria. And I also want to thank you for listening to my presentations. Thank you.
Thank you, Caroline. The study is quite deep, especially for us here. Most of our students here are undergraduate, right? Some master students or even PhD student. Yes, and our PhD student is also working on resistant, resistant pesticide. Because we are from plant protection.
Yeah,
who applied a lot of pesticide. So,
which could then induce the varieties.
Yeah, actually antibiotic or pesticides is also maybe in the same operon of the resistant, resistant genes.
That's why I'm looking so forward to see the genetic environment of the antibiotic resistant genes to really see to which genes they are associated with, and if there's larger like resistance cassettes that can be moved around.
That's right. Yeah. Okay, it's interesting, and it is actually really important for us and for you as the young generation. Yeah, your vision should be about that. Yeah, to study more details, to solve any problems. Yeah, for the environment and for the health and also food. Okay. So, any question? Any question? Boleh dalam bahasa Indonesia. Nanti kita terjemahkan kalau agak yang positif nanti dulu Pak yang undergraduate. Ini paham nggak? Tapi tak lihat kayak tadi itu ada yang mainan HP sendiri, ada yang ada juga sih yang serius, ya. Paham nggak ini? Paham nggak? Halo, ini semester berapa ya Mbak? Undergraduate-nya? Semester 6? 5? Wow, 5 udah selesai nih, 5 tahun. 5 semester, ya. Maksudnya sudah cukup banyak belajar, sudah 5 semester, sudah banyak ininya. Ada yang mau tanya? Ada yang ingin dijelaskan lebih jelas? Mungkin ada yang tadi apa sih gitu? Oke, silakan. Mau tanya? Kan gitu loh, udah jauh-jauh didatangkan ya kolega dari negara yang lebih maju, lab yang lebih berkembang, nggak suruh bayar loh, mereka bayar sendiri. Kenapa nggak dimanfaatkan untuk belajar? Ayo. Nah, nanti dulu kamu masih sudah hampir masuk postgrad. Mana yang ada grade dulu? Hah? Ayo, boleh silakan. Tanya apa? Oh, itu kelihatannya kemarin-kemarin ada. Saya itu pengen sekali gitu. Belajar dengan dari yang ribu loh. Enggak mau. Ini jadi bukan desainnya atau mentornya. Teman? Atau yang di online? Anaknya kamu cek ada yang di online? Mas Agus, ada yang di online ya?
You can also ask if you didn't understand something, like,
Yeah.
because I know these are really difficult subjects, and yeah, it took me time to learn this also, and they're a little bit confusing to me also. So no worries, there's no stupid questions, so you can ask anything.
Okay, and they are from the class of biological component. Very match with this topic. Gimana? Nggak ada yang nyantel? Nggak ada yang jendel? Kita luruskan. Sama? Wes, bahasa Indonesia wes. Jadinya kayak gini nih. Tiap kali. Nggak ada? Ya sudah. Tadi Mas Azmi dulu apa Mbak Nurul dulu ya? Ya Mbak Nurul dulu. Ready first. Silakan Mbak Nurul sini.
Okay. Thank you, Taro, for your presentation. It's such an interesting discussion today. I want to ask some questions. First, why did you decide to collect sample from manure too? What's your why did you decide not from only soil but from manure too? And then I want to confirm the process from start to the end. So we collect sample from soil and manure, and then we grow in media, and then we extract and do whole genome sequencing, and we align. Is it right? And we align, and then we do the anti-smash for detect gene cluster for which one that express antimicrobial anti-resistant gene, and then we apa namanya?
I can't do that.
Move so fast.
It's a little bit slow that.
Okay.
It's going to the naturalist, yeah.
Okay, and then after detect gene, and then we try to know which mobile genetic element that available in your isolates, and then we compare with ancient isolates. Is it right? I want to confirm the process from start till the end. And then the third question is, we know that in the isolate or in your isolates, is there is plasmid or not? Yeah. But okay, but can we know the specific mobile genetic element, others mobile genetic element beside plasmid? Tadi tadi ada plasmid, intron, transposon. We know that there is plasmid or not, but how about the other mobile genetic element? Okay.
Yeah.
Okay. Thank you.
So I myself, I did not do the isolation of the bacteria from the samples, and so they were already in front of my supervisor before I started doing my thesis. But if I have understood correctly, their interest because they are known to include also those pathogenic bacteria and the antibiotic producing bacteria. So it's interesting to know the interactions also between those two. So if there's antibiotic producers present that have resistant genes and the mobile genetic elements, it could be studied that are they also transferred to the pathogenic bacteria present in the samples. And also, I know those samples were part of the bigger projects which studies antibiotic resistance in those kind of samples. So my thesis and this antibiotic producer is like some smaller path from the bigger study. So, and did I remember to say, do you have something to add?
Yeah, like your samples were fertilized.
Yeah.
Yeah.
Yeah. Yeah. And the second question was about the process. So yeah, if I start from here, so yes, the bacteria are first, you have the samples, then we use one XPS where we put the samples to have like it's liquefied, so then it can be pipetted to the plates from dilution, and then we can see everything that, or well, not every bacteria that grows from the sample, but some bacteria. And then we choose the isolates or the colonies that look interesting to us. And yeah, we are quite cute on recognizing streptomyces that we are interested about. So then from the colony, from the mixed culture plates is cultures to new plates to have the pure pure culture. Yeah. And from the pure cultures, the DNA is extracted. And then the extracted DNA can be, for example, sequenced with this BacBio, but of course other sequencing methods can be used also. We chose this long-read sequencing because we want to also study the genetic environment of the resistant genes, although I did not do it in my thesis, but in the future it can be done from the long-read sequencing. So yeah, then when you get the sequences from your from the sequencing, they are like really of these short or raw reads. So then they are assembled into contigs, and these contigs are then analyzed with different programs, and the programs you choose, it depends what you are interested about the genomes. So yeah, we were interested about the resistant gene, mobile genetic elements, plasmids, and what they produce, because yeah, we want to know those. But of course, there's like so many options you can do while you have your sequences. And for example, from the sequence, this FACTA would be used. It shows you like every known protein coding genes that is present in your sample. So yeah, I saw like, okay, they might produce cellulase, and like stuff like that if you are interested about that. So yeah, the process really depends on what you are interested. And about the plasmids, so yeah, I did not detect any plasmids in my isolate sequences, but yeah, there still could be. Many of the plasmid database mainly includes like the clinical isolates plasmids and not the environmental bacteria, so the antibiotic producers. So yeah, we also should practice open science and download our sequences to databases, so the amount of environmental bacteria would be increased, and their genes and their plasmids and other. And yeah, we from the mobile genetic element database, we also see the mobile genetic element type. Did I answer to your all questions?
Yeah.
Okay,
Thank you so much. Okay, good. Okay, thank you, Tarun. And next. And maybe next, Mas Afif.
Okay, thank you. It's very interesting, but yeah, it's
Yeah,
difficult for me.
you can use
It's okay. Can you
Huh?
start?
Yeah.
No speaker mode.
like it.
Yeah, but I have a question. The first question is related to the manual fertilizer use. What the condition of the manual fertilizer can cause or trigger the most effect for antibiotic resistance? It's the very raw fertilizer or something that's very mature. The first question. The second, in Indonesia, we have an issue with some bacteria like bacterial wilt and some things where our farmer using the not only manure but also the organic fertilizer. From your theory and something, you know what is the effect for the unorganic or organic fertilizer cause the antibiotic resistant or related to increase the pathogenicity of the bacteria level of the virulence.
So, I don't know how these fertilizers has been processed, as they are from some company, so they don't really tell us how they produce them. But we advise that the big manure slurry at least goes some form of high temperature composting. But yeah, we don't know in detail. And also the BPF45, so the composted pile waste, it's not manufactured anymore, so we can't find it out. And yeah, from the problem of Indonesia using also unorganic fertilizers. So yes, it has been seen that like metals can make the antibiotic resistant genes also to be associated more with the mobile genetic elements, even more than the use of antibiotics. The metals can spread the resistance, at least in some study I read. So yeah, it's really a problem if you use like metals. And yeah, we were thinking, because we found out that some EM4 is used really much, and there's also some metals present. So yeah, it would be interesting to see that if the use of it can also like increase the resistance. Yeah, but yeah, we are still studying this. Thank you.
Okay, I think from Carol's presentation, it was also mentioned that when we use manure, that manure maybe come from animal with a high application of antibiotic. Yeah. And then you also found more resistant genes in the community, right? In the population. So of course, when we also add any other chemicals, including the unorganic fertilizer, that may be becoming more complex and probably also increasing the population of antibiotic or antimicrobial resistance.
I think this study I put here is quite nice about like they studied how the adding of antibiotics and metals and some other what is the effect in the in like the big manure. So yeah, it's a really nice study.
Ya, biasanya organisme itu kalau stres itu malah muncul yang tidak-tidak, semakin crazy. when we face any stressful things, then maybe many crazy gene expression will be.
Stress response.
Stress response. Jadi sama aja, tidak hanya kita ya. Kalau lagi ujian mungkin jadi pemarah dan sebagainya, gitu ya, namanya stres ya. Sama dengan mikrobioma dan kemudian tidak hanya supres, malah kemudian jadi aneh-aneh, ya, termasuk menghasilkan ini gen-gen resisten terhadap cekaman tadi, cekamannya antibiotik, cekamannya organic fertilizer, cekamannya pestisida dan sebagainya. Itu kemudian akan muncul resistensinya. Dan itu menimbulkan masalah baru. Tidak hanya masalah kita di food production, tapi juga health dan environment. Begitu mungkin. Ini Mas Azmi terakhir mungkin, atau masih ada yang mau bertanya? Iya, nanti dulu ya. Aku udah sedikit nangis. Soalnya penting ya. Terima kasih kita ya. Silakan.
Thank you for the presentation. It was a good presentation. So my question is, be a little detailed. So from the presentation, we know that there is GC content in genome of Actinobacterium mentioned twice. So my question is there any relation between GC contents in the Actinobacterial genome to the ARGs? If it exists, then how is the relation? So my second question is, if MTE sequence from Actinobacteria contains ARG and has moved through the Proteobacteria, and it's a transposon, is it possible to move to another sequence in Proteobacterial genome and reverse the trait back to susceptible, since we know that transposon can be moved unpredictably? Thank you.
Thank you for your questions. So, I forget the first one.
Is content related?
Yeah, the ARGs. In my understanding, the GC content is not related to the ARGs. Yeah, of course, depending on the GC content, there can be differences, but proteins are produced from the DNA sequence, but at least I have not come across any information about it would be related to the antibiotic resistance, but yeah, about the GC content, it can makes the bacteria to grow in more higher temperatures, or yeah, I think it's something to do with the different bonds of the G running to cytosine band compared to the others. But yeah, I'm not so expert about this. Thank you for the question. And the other one was, sorry, I'm starting to forget.
If an MTE sequence from Actinobacterial genome has moved and it contains ARGs and moved to the Proteobacteria and it's a transposon, is it possible to move to another sequence in the Proteobacterial genome and reverse the trait back to
In my understanding, yes, because then if this carryback model has happened, the Actinobacterial gene is inside the Proteobacterial DNA, so the Proteobacterial DNA genes will recognize those, and then it can be moving around there. And also, Proteobacteria are able to transfer their genes to other bacteria by the conjugation. So yeah, then it can also be spread again to Actinobacteria and other Proteobacteria. So, thank you.
Okay, anyone else? Please. Silakan, yang mulai mau tanya. Ada lagi? Ada? Oke. Asmi?
Oke.
Tolak terus sama ibu.
I have two questions. The first is, you use ISP6 and ISP7, but the culture only grow in ISP7. Why? Only ISP7. And what is the difference between both of them? And the second, all of your samples no contain any plasmid, no contain plasmid. As long as we know that the gene can lose when the gene, especially RGs, can lose when there is no selective pressure or there is no RGs. I mean, there is no antibiotic in the media. So, how can you make the gene not lost in your sample? Because sometimes we can lose the gene, especially RGs, when we grow the bacteria in the media that not contain any antibiotics because the burden on the gene itself. So, thank you.
Thank you for your questions. So I think one of our problems with the ISP6 was that it was expired. It was really old. So at least when I saw pictures in other articles, the streptomyces on it looked like totally different. Of course, different species, different looks, but yeah, I didn't see in any other like studies that streptomyces would look like that they look on my species. So yeah, it can be because my species did not work properly, or well, they are different species and strains, and that's why they go on the ISP6, ISP7, but not ISP6. And yeah, there were differences between the ISP6 and 7, and I'm trying to find what is the. Do I have some? Yeah, I can share this slide. I'm not so good with. Yeah, I can share it here. So in here you can see the yeah. So there is different. So in the ISP6, there are like some peptide digest of animal tissue and proteins and peptone, and the ISP7 they are there's like tyrosine. But yeah, I'm not so expert what the different ingredients on media do. Yeah. And the other question was,
how can you maintain your gene in your media?
I don't know the answer for that because yeah, I have also read that especially streptomyces can mutate during culturing and yeah, lost their genes or stuff like that. So yeah, I don't know unfortunately the answer.
I can also comment that I think they have generally they are different in their environment than in the place, like every bacteria, because it's a different environment and no competition. Yeah, so it's not the same, but it's just a rough idea.
Well, the metagenomic sequencing is nice, and then you can do sequencing the whole sample and see like maybe in more detail like every genes that are present. But of course, then you don't know who carries them. But maybe we will overcome this problem in the future, and yeah, we need to study it.
Okay.
Find a way.
Yeah.
Thank you.
Many things. Yeah, that's to be studied. Yeah. Actually, there are many interesting things that we haven't done for studying. Mantap. Yeah. So, very good. And I think there are more students coming, but you are late, especially there. You're working on streptomyces. Okay. Anyone else? Any questions? Yeah. No more? Berapa lama ini? Baru 1 jam 15 menit. Masih banyak. We still have some time. Silakan, ada yang tanya lagi? Bahasa Indonesia nggak apa-apa. Sudah terpancing tadi dengan penjelasan-penjelasan yang ditanyakan. Atau dia punya pertanyaan, biar di-share. Oke? Sudah nggak ada? Sudah. Silakan Mbak Nadia, itu tadi pembawa acaranya. So, Thank you very much again. We give applause for Tarun. Study. Thank you. Okay.
Ya, terima kasih untuk Prof. Siti Fadia telah memoderatori acara hari ini, dan terima kasih untuk Taru. Thank you for your presentation. Dan jika kalian tertarik dengan tema hari ini untuk mau join dengan proyek, monggo silakan menghubungi Prof. Siti Fadia. Mungkin mau tertarik dengan antimikrobial resisten ataupun resistoma, resistoma. Ya, I'm sorry. Monggo silahkan menghubungi Rofia, jadi masih bisa diterima.
Penggunaan pupuk-pupuk tadi loh, pupuk organik. Ternyata itu tidak hanya untuk memecahkan masalah, tapi terkadang juga menambah semangat. Itu, jadi itu banyak yang perlu diteliti lebih lanjut. Ya.
Iya, masih banyak lahan penelitian yang bisa kita gunakan ke depannya, mungkin anti pestisida, anti insektisida, dan lain sebagainya. Dan terima kasih untuk hari ini. Jika mungkin ada pertanyaan lebih lanjut, bisa langsung menghubungi Prof. Siti Fadia ataupun langsung ke Taru. Dengan nanti kita share PowerPoint-nya. Dan terima kasih sudah menghadiri acara hari ini. Ya, ini adalah merupakan guest lecture terakhir untuk tamu dari Universitas Helsinki, dan untuk kedepannya kita ada kedatangan tamu dari Jepang, yaitu Prof. Toru dari Jepang. Ya, nantinya akan memberi guest lecture juga mungkin di hari Kamis, besok Jumat, di hari Jumat. Kalian mungkin bisa menghadiri kembali karena masih ada banyak pelajaran yang kita bisa ambil dari beliau gitu. Dan terima kasih hari ini. Wassalamualaikum warahmatullahi wabarakatuh. Ya, mungkin bisa foto bersama di depan lift seperti biasa. Jadi silahkan memposisikan diri.